Hello, everyone. Thank you for joining us today for our ALS Webinar Wednesday series. My name is Alejandra Herrera, and I'm the Marketing Coordinator for ALS North America. I will be facilitating the webinar today. If you're having technical issues with our webinar platform or have any questions, please use the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenter today is Allison Nito. Allison is the current biology manager at ALS Environmental in Winnipeg, Manitoba. She graduated from Lakehead University with an honors Bachelor's of Science in Applied Biomolecular Science and has worked with ALS for 15 years. Today, Allison will be discussing SARS-CoV-2 analysis in wastewater and swabs. Allison, I will now turn it over to you.
Thank you, Alejandra. Hello, everyone. I'd just like to start off by thanking you all for taking time out of your busy schedules to join us today for this webinar. As Alejandra already said, my name is Allison Neto, and I am the biology manager here at ALS Environmental in Winnipeg, Manitoba, Canada. Today, we'll be discussing SARS-CoV-2 analysis in both swabs and wastewater, look at the benefits of testing, what's involved in sample collection, give a brief overview of the PCR technology, and also take a look at the generated report. I'll open the floor at the end of the presentations for some questions as well. So I'd like to start by just giving a brief overview of the types of services that we provide at ALS. Our scope is very diverse, and we're able to provide a wide variety of analytical services on matrices, including soil, water and wastewater, air, environmental samples, and food. Our services include environmental chemistry, microbiology, toxicology, industrial hygiene, food safety, as well as wastewater analysis, provided over approximately 126 locations worldwide. Locally in Winnipeg, our capabilities include organics analysis, which involves tests such as BTEX, THM, oil and grease, GC volatiles, and GCMS volatiles. Inorganic analysis for ICP-MS metals, mercury, major anions, cyanide, chlorophyll, and reactive silica. Our limnology section, where we test industrial hygiene parameters such as asbestos and radon, mold, spore trap, benthic and algal ID, invasive species, and microtox. And finally, our microbiology section, where our tests range from coliform testing, HPC and Pseudomonas, to food testing, ribotyping, Cryptosporidium giardia, Legionella, and finally PCR. Our ability to perform PCR testing provided us with the framework to undertake our newest analysis, which is SARS-CoV-2 or COVID testing in both swabs and wastewater. The first confirmed case of SARS-CoV-2 in the US was documented on the 21st of January of 2020. Within a few weeks, a global pandemic was declared, and the end of 2020 would see over 83 million cases and almost 2 million deaths globally due to the virus. The combination of rapid spread, human transmission, and exponential growth in cases provided the impetus for scientists worldwide to gather as much information as possible in hopes to control the massive spread. But what exactly is SARS-CoV-2, and how is it able to cause such widespread issues globally? SARS-CoV-2 is part of the coronavirus family. Now, these are large, roughly spherical particles with unique surface projections, causing respiratory tract infections in humans. The spikes are the most distinguishing feature and are also responsible for the corona or halo-like surfaces, hence the name coronavirus. S1 proteins are the most critical components in terms of infection, and they possess two major domains, which are the N-terminal and C-terminal, which serve as receptor binding sites. These sites recognize and bind sugars on the surface of the host cell. Infection begins when the viral spike protein attaches to its host cell receptor. And after this attachment, a protease of the host cell cleaves and activates the receptor-attached spike protein. Depending on the condition, the virus can either enter the cell by endocytosis, where the host cell surrounds the virus membrane and ingests the foreign material, or by direct fusion with the host membrane. Now, there are two main modes of transmission by which SARS-CoV-2 can potentially infect humans. The first is one that we all know about, dealing with mask mandates in various regions, and that's aerosols. Now, these can spread from an infected person to others through respiratory droplets, or they can be created when an infected person coughs, sneezes, sings, shouts, or talks. Droplets can vary in size from very large droplets that fall to the ground rapidly near the infected person to smaller droplets, which can linger in the air under some circumstances. Now, the virus can also spread when a person touches another person, surface, or object that has the virus on it, and then touches their mouth, nose, or eyes with unwashed hands. These surfaces can include items such as doorknobs, faucet handles, toilet levers, stair rails, light switches, and they're collectively known as fomites. Now, this is one of the driving forces behind the enhanced cleaning protocols that have been adopted by various workplaces, schools, and businesses in an effort to help disinfect and limit the spread. Now, given what we know about fomites and the role they play in the transmission of SARS-CoV-2, what can we do to ensure that our enhanced cleaning protocols are effective? Now, this is where swab testing at ALS can be extremely beneficial as part of any COVID monitoring plan, or in the worst case scenario, any remediation plan following a potential exposure or positive case within your facility. So this testing helps to protect by identifying which surfaces in a given facility may play a role in the onward transmission of COVID-19. Common surfaces that may be considered in such a monitoring program include time clocks, restrooms, carts, doors, handles, lunchroom tables, and chairs. Upon requesting analysis, ALS will provide all necessary materials required to sample wastewater and/or swabs, including a shipping cooler, ice packs, chain of custody form, and a waste bag to dispose of all leftover sampling materials safely. Sampling instructions can also be provided by the lab, and we'll touch on that in the next slide. Now, for swab testing, up to 3 separate swabs can be pooled at the time of sampling to create one composite sample for testing in the lab. So this can help you as a client to test more areas within your facility while still helping to keep the program cost effective. There's a recommended procedure for actually taking the swab sample, which is available to clients upon request. So before any sampling begins, ALS does recommend that you follow all of your local protocols in terms of appropriate sampling PPE. Now, the first step in the process is to remove the swab from its sterile wrapping and moisten the tip of the swab in the tube marked saline solution. You're going to gently press the swab on the inside of the tube to remove excess moisture and swab the surface to be tested while the swab is still wet. You're going to place the moistened swab on the surface and streak across the surface, rotating the swab between the thumb and forefinger. Now, for flat surfaces, we recommend to rub 10 times horizontally and 10 times vertically in an approximately 5 by 5 centimeter area and avoid letting the swab dry. Once you've swabbed the area to be tested, place the swab inside the storage solution tube. There is a preservative in the vial that lyses and inactivates the SARS-CoV-2 virus if it's present, which helps to render it non-infectious. As a result, sample shipments to the laboratory really have very little potential to be dangerous. So you're going to break the shaft of the swab by pressing it against the tube, and you're going to seal the tube slightly, disposing of the broken shaft in the waste bag provided. The sample ID, or how you want the sample to be identified on the report, is recorded on both the storage tube label as well as on the chain of custody. And you're going to place that sample storage tube in the bag provided and disinfect the outside with the provided alcohol wipe. You're going to place all the samples, the completed chain of custody, and frozen ice packs in the cooler, tape shut, and prepare the cooler for return shipping. Now, there are some important items to consider before shipping your samples. Your sample tubes must be kept refrigerated, both prior to use as well as after sampling. As mentioned above, an ice pack will be provided to help you to maintain cold conditions during shipping. You want to ensure that the sample ID and description are recorded on both the chain of custody form as well as on each individual swab. And the information recorded on each of these 2 items must be legible and they must match. So ideally, samples would be received at the lab within 24 hours of sampling. However, analysis can be completed up to 72 hours after sampling. If you need to sample and ship the sample over the weekend, we can provide that service here in Winnipeg. However, we do strongly recommend that you contact the lab first to ensure that someone would be here to receive and analyze the sample as required. We usually are able to accommodate emergency requests, but we do need to know about them ahead of time. If you do not make these arrangements, there is a risk that your sample may exceed the recommended hold time before the analysis is performed. Once the lab has received your samples, you'll receive a sample receipt confirmation, or an SRC. So this confirms to you that the sample has been received, what the expected delivery date of the report is, as well as provide you with the unique sample identification number assigned to your sample. ALS also provides WebTrieve access as a value-added service, where once you're registered, you'll be able to see the status of your sample, view results, etc., via the computer or mobile app. So now I'd like to shift our focus slightly to wastewater surveillance and analysis. So the wide application of wastewater surveillance began in the 1990s with efforts to eradicate polio. As large-scale vaccination reduced polio transmission, the conventional approach to monitoring polio, which was tracking cases of paralysis, proved incapable of predicting and preventing isolated outbreaks. Now, because poliovirus infections often present with nonspecific symptoms, and with paralysis occurring in approximately 1 in 200 cases, the virus was able to spread undetected in areas where it was thought to be eliminated. Testing sewage for poliovirus RNA proved 4 to 5 times more sensitive in detecting outbreak than monitoring communities for an increase in cases of paralysis. It also allowed entire communities to be closely monitored. Now, this theory is being applied in the fight against COVID-19 and being utilized globally. In terms of COVID-19, wastewater surveillance is based on the theory that infected individuals can shed the virus in their feces as soon as 3 days after infection. Since the shedding would occur before the individual would become symptomatic, monitoring COVID-19 levels in wastewater could provide public health with vital information as to potential hotspots or a potential increase in cases much sooner than conventional individual testing approaches. Knowing when outbreaks are declining is also useful information to help prepare for things such as pandemic recovery or reopening plans. Now, this graphic is from a team of Saskatoon researchers who were able to predict a surge in cases at the end of 2020, approximately 1 week in advance, using wastewater surveillance monitoring. It's a very cost-effective way to survey transmission dynamics of entire communities while avoiding the inherent bias of other epidemiological indicators. And those biases include things like collecting data for people who may lack access to healthcare, and including information from people who have very mild symptoms or who are asymptomatic carriers that would likely not go and seek a conventional test. It can also provide public health officials with near real-time information on disease prevalence. It would mimic testing an entire population at once at potentially frequent intervals, something that could not be accomplished with clinical specimen testing. Experience with other viral diseases, such as polio, has shown that monitoring sewage for traces of pathogen enables effective surveillance of entire communities, providing a sensitive signal of whether the pathogen is present in the population and whether transmission is increasing or declining. Now, if you're interested in establishing a wastewater surveillance program at your facility, it may be necessary to review construction drawings or engage a consultant to determine optimal location or locations and your testing frequency. Accessibility, security, and power availability are things that all need to be considered when developing this plan. Wastewater should be collected using a composite autosampler over a time period representative of the population profile. So generally, there are 2 recognized types of composite samples. The first is known as a time-weighted composite sample, and that requires the collection of multiple sample aliquots of equal volume collected at a consistent time interval over a specific period of time. Now, a flow-weighted composite sample is the more reliable and preferred method of composite sampling because not only is it collected over time, but it's also proportion based on flow volume. So when flow-weighted composite sampling is performed, there are many more sample aliquots taken when discharge is flowing at 200 gallons per minute, for example, as opposed to when only 10 gallons per minute are being discharged. So this sampling method results in a more representative composite sample as compared to a simply time-weighted sample. Now, for example, if you're running a 24-hour operation, it should be sampled over a 24-hour period. Whereas if you're operating a site that is using an 8-hour workday, those are the hours when samples should be taken. So again, you want to make sure that you are capturing when the most people would be using the system. Sampling points for actually taking the samples should be prior to any treatment, and they're selected to capture the wastewater outflow from the population to be monitored. Sampling frequency should also be based on monitoring objectives and risk profile. And daily composite samples are recommended. However, factors such as local community viral spread statistics should also be considered. And again, we do recommend contacting a consultant to help determine frequency and sampling plans, as this is not a service provided by ALS. Once the water has been subsampled into a 250 milliliter plastic bottle, samples should be chilled and expedited to the laboratory within 72 hours using the provided package with ice or ice packs. You can use all of the same protocols that we discussed for the shipment of swabs to the lab, as they also apply here. Ideally, packaging must maintain refrigeration temperatures, so 2 to 8 degrees Celsius, and the samples should arrive to the laboratory within 72 hours from sampling. Once samples are received in the lab, the analysts prep them for testing. While both swabs and wastewater samples are analyzed using the same PCR technology, the process and kits used for each are slightly different. So that's why you'll notice a difference in turnaround times, methodology, reporting, etc. However, the PCR process is generally the same for both matrices, so we'll just briefly go over a general overview of that process in the next few slides. So for swabs, the sample is first processed for nucleic acid extraction and amplification. Since SARS-CoV-2 is an RNA virus, the first step is to convert that RNA, if it's present in the sample, into DNA using a process called reverse transcription. Primers, which are specific gene sequences connected to enzymes that copy DNA, are used to amplify the target DNA in the test sample extracts. Amplification of DNA involves heating the samples to separate the DNA strands, duplication of DNA using specific primers and enzymes, followed by cooling to rejoin the DNA strands. This cycle is repeated multiple times and is called polymerase chain reaction, or PCR. Probes attached to fluorescent markers are used to bind with targeted DNA, which makes the amplified copies of the virus detectable by PCR instrumentation. If the amplified fluorescence level exceeds a predetermined background threshold, which is also known as the cycle threshold, the test is considered positive. Both positive and negative controls are utilized throughout the test as routine quality control checks to ensure the validity of the test results. A positive result would indicate the presence of SARS-CoV-2 genetic material. If the targeted genetic material was not present in the sample, the PCR process would not have made any copies and the test will be considered negative. Now, the PCR theory for wastewater is the same. However, this analysis is quantified. So wastewater is analyzed using RTPCR, which is a reverse transcription polymerase chain reaction test that both detects and quantifies RNA from the SARS-CoV-2 virus in untreated wastewater. When samples arrive at the laboratory, they're pasteurized at 60 degrees Celsius for 1.5 hours in order to render the virus inactive. As you may recall from our previous slides, this would be the same theory behind using the storage buffer for swabs, so safety and protection from potential exposure. Now, before the concentration step can begin, it's important that the sample be brought back down to a temperature of 2 to 8 degrees Celsius in the lab, again, in order to prevent sample degradation and ensure efficient precipitation of the SARS-CoV-2 virus. So this is why you'll see a little bit of a difference in the turnaround time for this test, as there is the extra pasteurization that is not required with swabs. Now, the next step in the procedure would be to concentrate the sample using a combination of centrifugation and polyethylene glycol sodium chloride precipitation step. The RNA is extracted from the sample using magnetic bead separation, and then the genetic material isolated from the sample is amplified using the SARS-CoV-2 real-time or reverse transcription PCR test using the same mechanism as described in the previous slide. Now, after the PCR analysis, the two analytes diverge in terms of reporting. The swabs are reported qualitatively, while the wastewater samples are reported quantitatively. And when we consider what the end use of the data would be, this makes a lot of sense. So when a client is mounting a wastewater surveillance program, they would need to have a finite result in order to plot the data and observe trends over time. In cases where a client may be verifying a cleaning protocol with a swab, routinely monitoring a workspace, or remediating after potential exposure, any detectable presence of SARS-CoV-2 indicates a problem and would likely trigger another disinfection. So for this instance, the quantified presence of SARS-CoV-2 is less significant. Now, in an RT-PCR assay, a positive reaction is detected by accumulation of a fluorescent signal. The cycle threshold, or CT value, is defined as the number of cycles required for the fluorescent signal to cross the threshold to reach a detectable level. CT levels are inversely proportional to the amount of target nucleic acid in the sample. So the lower the CT level, the greater the amount of target nucleic acid in the sample. Cycle thresholds of less than or equal to 29 are considered strong positive reactions, indicative of abundant amounts of target nucleic acid in the samples. CT values of between 38 and 40 are very weak reaction of the target nucleic acid. Now, this is a mechanism that the analysts look at in the lab when we're evaluating the data, but this isn't actually a reported parameter that you would see on your final report. Quantification is then performed using a standard curve produced using the appropriate nucleic acid reference material. The standard curve is created by diluting reference material in a series of known concentrations and then measuring a value for each concentration. The unknown samples are then compared to the standard curve and a value can be extrapolated. The calculated relationship can then be used to convert a CT value measured for an unknown sample into a concentration of RNA expressed as genome copy per volume. Now, once the analysis has been completed, you will receive a report that looks like this. So you can see that your report will have a unique laboratory identification number, which is here. And you will have also got that number on your sample receipt confirmation that we spoke about earlier. And also it will have the sample ID that you indicated on your chain of custody. It will also indicate the matrix that was submitted, so whether or not you submitted a swab versus water. And as well as the parameter tested, so in this case, SARS-CoV-2. The second column here will show the result that we got for the test. So in the first sample detected, because we analyzed a swab, and a result of 110 because we analyzed water for the second one. And the third column is for qualifiers. So your test report may or may not have anything recorded in this column. So this is for communicating information to the client that may have affected the test. So for example, things like exceeded hold times or an incorrect sampling container that may have been used would be communicated here with a legend at the end of the report telling you exactly what each qualifier means. In cases where the hold time is exceeded, we will call you and get your permission to proceed with analysis. However, you will still see the qualifier on the report. The fourth column shows the unit of measure. So again, it's blank for the swab sample since we're using a presence-absence method, and shows a unit of copies per milliliter for the wastewater sample. And finally, it shows the date the sample was analyzed in the lab. Now, the final page of your report will show you the ALS test codes, as well as providing you with a method reference that the lab is using, as well as a brief summary of the method as well. This page will also have information regarding any qualifiers that may have been present on your report. And now the above slide shows laboratory turnaround times as well as list pricing for both swabs and wastewater. However, if you are considering operating a large scale project, we do recommend that you contact the lab and we would definitely be able to work something out for larger scale projects. And that basically brings us to the end of this webinar. So should you have any questions regarding testing or about your final report, please feel free to contact myself or the laboratory manager, Barbare, at the emails provided on this slide. And as well as there's the general laboratory phone number as well. So I guess I will turn things back over to Alejandra.
Thank you, Allison. Before we begin the Q&A, I would like to ask everyone to take 2 minutes to complete our survey. The link is in the chat section. We would love to hear your feedback. Okay, we will open the forum for questions now. Please write out your questions using the Q&A function in the toolbar at the bottom of this webinar. I will give everyone a few more minutes to type your questions. In the meantime, I will review some brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS Global website and our ALS YouTube channel in the webinar playlist. Also, please follow our company page on LinkedIn as we post announcements and registration links to future webinars and other valuable resources and updates. Okay. Mary is asking, would it be possible to test surface water for COVID if there's expected septic seepage, or would that be too low concentration?
So when we're dealing with a PCR analysis is designed to take samples where you might have a very small amount of material and amplify it in order to be able to quantify. So you're making many copies. So the low quantity of the virus in the sample wouldn't really affect the PCR. If you're having seepage, what you might see is an interference when we start running the PCR based on the kits, et cetera, that we're using. But that would be something that we'd have to evaluate on a case-by-case basis and let you know if we did see an interference at all based on something else in the test sample.
Thank you. Pamela is asking, have you seen any trends of downstream transference from areas that spike with SARS-CoV-2 to further infection spread downstream?
Not in our experience with testing here at the lab. It's not to say that it couldn't happen, but it hasn't been something that we've encountered ourselves.
Do we have any more questions? What COVID PCR kit do you use?
We're using a COVID PCR kit marketed by IDEX.
We got a question from Jason. Could you estimate how long the virus could survive if, for example, a lagoon releases into a river, how long it lives in the river?
So viruses are very hardy. So essentially what a virus is it's just genetic material encapsulated in a protein. So what viruses are very good at doing is surviving in adverse environments until they receive until they can come into contact with a host or with an environmental condition that's favorable for their replication. I mean, theoretically, a virus in that state could survive, but would not be able to necessarily infect or replicate until the conditions were favorable.
Chris is asking, how eloquent is your PCR method with regards to detecting CoV SARS-2 variants?
We're running a detection limit of less than 8 copies per milliliter.
Okay, well, thank you, Allison, and thank you for attending. Oh, we have another question from Adam. Have you seen any effects on the wastewater COVID detention after treatment with chemicals such as ferrous chloride or peroxide?
This PCR test is actually designed for raw wastewater samples, so anything prior to treatment, as chemicals used in the treatment process can cause inhibition that we sort of touched on earlier. So the samples that we're analyzing for this method are all in fact raw and not treated.
Mark has a question. Do you normalize your results with anything? Pepper virus?
It's something that we're looking into at this point. The analysis here in Winnipeg is still in the early stages, so we are looking into using the mottled pepper virus as part of our assay.
Jason's asking, could you comment as to why the cities are not embracing this further across provinces?
Some of the feedback we're getting is that it's a very provincial approach. So there hasn't really been a federal approach to this in Canada. It's my understanding that in the US, I believe that funding for this is covered by the US government. So just the ability of the governments to provide some financial relief is putting an impact on it, especially when you're talking about smaller water treatment plants or smaller systems that may not have the funds to take on something like this. So I would say that that's probably the biggest factor at this moment.
Mark has a question. What volume you require being sampled, as if you suggest filtering to increase TSS in submitted sample?
We actually recommend that you submit to us 250 milliliters of sample. You don't need to actually do any filtration. The concentration for our method involves multiple rounds of centrifugation. So we're actually analyzing the supernatant and helping to get any of those particles out in the centrifugation process. So aside from actually sending us the water, you don't need to do any further treatments to the samples prior to submission.
Any more questions, anyone? Well, I want to thank everyone who attended the webinar, and I hope you enjoy the rest of your week.
Thank you very much, Alejandro. It was a pleasure spending this half hour with everyone today. And again, if you have any further questions that you may think of after the presentation is completed, please feel free to reach out.